This is how to properly assemble and use the extruder for the Liposome 202 lab. First, take two small filter support pads, ensuring that they're separate. Remove the larger filter pad from the two blue protecting patches on each side. Using forceps and a disposable pipette, dampen the filter support pad with PBS buffer and carefully place it within the O-rings of the internal membrane supports on each side. Place one of the finished membrane supports inside the outer casing. Dampen the main filter and add it on top of the membrane support in the casing. Add the second membrane support, sandwiching the main filter, then add the Teflon bearing and the retainer nut. Then, insert the assembly into the rack. When inserting the needle, ensure to put them in straight and flat. Poking the needle into the Teflon bearing can damage or bend the needle. To prime the extruder, pull a syringe of PBS buffer and insert it into one side of the extruder with an empty syringe on the other side. Slowly pull the PBS back and forth to prime the extruder, holding both syringes inward on the membrane to provide a tight seal. Don't be alarmed if some volume is lost inside the extruder. There's negative space to make up. Once the extruder is primed, repeat these steps but with the sample. To clean, pull the needle straight out and dispose of the sample or the PBS in the proper waste bin. Then, with both syringes, run a syringe of isopropyl alcohol through them, and then a syringe of DI water. Remove the plungers of the syringes so that they dry properly. Then disassemble the extruder, throwing away the filter and filter support pads. Spread them out to dry properly.